and this is the Human Enhancement Podcast with your host, Jeffrey Wu. And we're back with a renewed vigor with our first guest of the year, Ira Pastor. And I'm really excited to have this conversation because Ira has an impressive background being in the pharmaceutical world for over the last 30 years and has built quite a range of experience being plugged in on the pharma side, as well as looking at the future in human enhancement, being a member of the World Economic Forum in human enhancement, and other, I would say, community and thought leadership groups around the notion of human enhancement. I think that's always been a little bit of sci-fi, but folks like yourself and folks like us, we're working on making that sci-fi reality. So excited to kick off the new year with this conversation, Ira.
Great. Well, I'm very excited to have the conversation too, and thanks for having me.
Absolutely. So, it's interesting to hear your background story here. So typically, I would say with my conversations with folks in the pharma industry, very, very focused on medicine. I think that's a very rigorous, expensive, slow process. And I think when we talk about human enhancement, very vague, perhaps I would argue, maybe it started off less rigorous, but I think more and more technology and science is becoming, as rigorous as just straight pharmaceuticals or straight medicine. How did you get interested, or were you always interested in the notion of enhancing human performance?
Ever since a very young age, I joke that, growing up, I was a big fan of comic books and science fiction, and whether it was superheroes or people that were traveling around the solar system, I was always rather intrigued by the human possibilities. I grew up in a family business here in the Philadelphia area that was involved in community pharmacy, so it was sort of preordained that I went into that at an early age. I became a pharmacist and I went to business school. And then I spent the last, as you mentioned, a few decades in the more traditional pharmaceutical industry. And, it was a fascinating place to hang out. But at the same time, I got a little dismayed by the fact that in generating, $1 trillion a year, and spending 200 billion more on new R&D, we were having a pretty bad success rate at dealing with and curing the chronic degenerative diseases that are primarily responsible for our suffering, degeneration, and death.
Right.
So
you probably saw the recent news with Pfizer pulling out of Alzheimer's and Parkinson's research, right?
Yeah, I just had an argument online with somebody about this that said, well, that's a good thing. I said, no, this is a horrible thing, because it's one thing for a big pharma company to get out of an area that's not core anymore, or something that, we pretty much, painkillers or what have you. But when you have the fourth largest drug company in the world abandoning what is arguably probably the biggest problem we have now and is coming in the next 10 to 20 years, that's not a good sign.
Yeah, that's concerning, right? Yeah, absolutely.
Yeah. It's a problem.
Yeah.
It's a problem for everyone. So I wanted to do something a little different after the first 30 years. I was not content with just marketing the next cholesterol-lowering drug or non-steroidal anti-inflammatory agent, and I really wanted to put together. I know that the last 100 years has been full of fascinating research, and a very small percent which is ever translated into anything. I really wanted to dive back in, not just think about the future, but dive back into the past. And when I talk about the past, I talk about the 3+ billion years of biological evolutionary discoveries that exist on this planet. What I mean by that are the wide range of organisms that inhabit this little blue marble with us called Earth, which from a health and wellness perspective are much further advanced than we are, whether that be the organisms that exhibit complex forms of regeneration and are effortlessly able to regenerate limbs, spinal cord, complex organs like the heart, eyeballs, and even large parts of the brain. And simultaneously, we are very interested in the resilient organisms, the ones that get cancer and shrug it off as if it was the common cold, and don't die from it in huge numbers, like 8 million a year, like we do. And then lastly, of course, at the far end of the spectrum, as you're aware, there are organisms on this planet that do not age. There are those that age in reverse. And there are a few that technically, even die and are reborn. And so, needless to say, we as humans are very bad at doing any of this type of stuff today. We ask the question, what can we do to push these types of discoveries forward, this type of research, and not get caught up in the traditional pharmaceutical model of the single magic bullet, of creating those little pills that have a single compound that do one thing. Because we have to tell you, if your arm falls off, it's not going to regenerate with a single magic bullet. It's going to be a much more complex therapeutic intervention than we currently think of today from Big Pharma. So that is, in essence, sort of the basket of how all of this came together and why we decided to go down this path.
And that vehicle is called Bioqark, and that's focused on regeneration of tissue and cells.
Exactly. And it's a little more than regeneration is part of it. We call and we talk about the 3 R's: regeneration, which is the big sexy one. But then there's even the larger one, which we call reversion, which is basically the ability to take cell B that is damaged in some format, where you don't want a regeneration event, but you want a repair event. You want to erase the history of what was and go back in time and start over, whether that be taking a cancer cell and erasing the epigenetic and genetic damage that has occurred to create a cancer cell in the first place. And then lastly, the last R is the rejuvenation, and basically how we can take the understanding of how we reprogram and remodel cells, tissues, and organs, and use it for ultimately the big picture down the road and turning back biologic age.
So what are your most promising compounds or interventions in those areas?
Yeah. So just the background. So we are focused primarily on creating what are called combinatorial biologics. So simple term, if you think of any biologic product today, a therapeutic protein like a growth hormone or insulin or a vaccine. However, we are looking to mix combinations of biologics to affect more than one target. And the reason being, complex events like regeneration are not single target events. So we really need to think beyond the single chemical, single target. So we're developing biologics, and our lead candidate is something that we call BQA, also known as a bioquantine. And what we are doing in this particular case is taking a series of peptides and proteins that are found in ooplasm that are normally involved in the reprogramming dynamic that we see early on in the development of the human embryo. The human embryo is the only place in humans where you actually see a reversal of aging. All our children are born aged 0 when they're born, and they do not come out of the womb after 9 months with the chronic degenerative diseases of old age. You don't have babies born with Alzheimer's disease, for example. So we studied a long history, the last 80 years of ooplasm reprogramming dynamics, which have primarily been studied in the Petri dish for cloning purposes, and said, you know what, we are going to now access those moieties, and instead of using it as a Petri dish technology or a stem cell technology, we're going to look at therapeutic opportunities and how we can use these bioactive substances as therapeutics that can be specifically formulated in different delivery vehicles to either regenerate tissues.
So these peptides are, peptides found in human embryo that for whatever reason, age out in a fully grown adult, and we're figuring out here how to reintroduce those compounds back into an adult.
Exactly. The ooplasm research has gone on since the 1940s, but it's just one of those things that stayed in the Petri dish. I mean, John Gurdon in the UK just got the Nobel Prize for this in 2012, many years, decades after he made his original discovery. So things took time, and we're basically taking the next step and saying, look, this is fascinating work, but we want to get it out of the Petri dish.
So I'm curious how you engage with skeptics around, we're more complicated than a starfish arm. A starfish can regenerate a leg or even, completely clone themselves essentially in form 2. So, one, we could make the argument that, of course, humans seem to be a little bit more complicated than a starfish. And then two, the reason why we don't have all these undifferentiated cells ready to differentiate is as a way to stop cancer, right? If you have undifferentiated growth that goes out of control, that's essentially cancer. So how do we overcome sort of nature's natural inhibitions on regeneration?
You bring up an excellent point, and I'm going to throw something back at you just because this is sort of you brought up the double-edged sword of regeneration and cellular proliferation, but the most fascinating point is that the most cancer-resilient organisms in the world on this planet are the regenerators. And there's a fascinating paper, it was published out of Tufts University a couple of years ago about regeneration. Is it the cause of cancer, or is it actually a potential cure? Because you take a regenerative organism, any regenerative dynamic, and this goes for not just the parts of organisms that regenerate, but this is also shown in mammalian embryos. You can stuff it full of cancer cells, and when the baby is born or when the limb regenerates at the end, cancer is not there because the microenvironment reprograms the tissue and has this wonderful capability to organize in what is needed and out what is not. So what we have, the direction we're coming from, and it's a great thing you bring this up, because a lot of people along this path that have sort of gone this way in the past have said, well, let's just find out what the genes are. And then we'll do some genetic engineering and we'll turn up those genes and everything should be hunky-dory. But that's not the case. Because what you do when you dive in, you find out what are all the genes that are active. Majority of them are oncogenes, right? Because they're the same genes for continual proliferation. So the answers don't have to do with how we stimulate large amounts of the regenerative substances per se, but more on the genomic architecture that controls them. And basically how those genes are turned on and off in the right tissues at the right time. So yeah, we're not interested in uncontrollable regeneration, and we don't want to turn you into a starfish or a planarian or a newt. But keep in mind that genetically, we're all pretty similar. It's one of the things the Human Genome Project showed us 20 years ago is there's not much difference in the genes. The difference they found was in the architecture around the genes. And this, in our opinion, and that is in essence what you find now when you shift.
So when you mean the architecture, you mean like the epigenetics, you mean the peptides floating around in the substrate, or exactly. DNA is floating around, how these things are unfolded, what's being expressed. Okay.
The epigenetics, the chromosomal architecture, one step above that, the biomolecular architecture of the intracellular fluid, of the biophysical dynamics of reaction diffusion within a cell, which then takes their cues from the tissue level. It goes a long way. I talk about these vertical hierarchies. So I am not a huge fan of reductionism in biology.
Yeah, bio is complicated.
Biology is very complicated. The pharmaceutical industry made a huge profit the last 100 years on reductionist thinking and breaking everything down to the smallest level. But hey, as we say, necessary but not sufficient. Our genes are definitely necessary, but they're by no means sufficient to describe how Jeffrey Wu or Ira Pastor becomes who we are. There's a higher level of architecture there that is responsible for that. And so unless we pay attention to those levels and how they communicate with one another, on up to, the biodynamics of the extracellular fluids and the extracellular matrix, we don't have answers to these problems. And so that's why we're taking, I don't use the word that much, but much more holistic view on biology. I think a lot of the systems bio people really are going in the right direction with this.
Absolutely. It seems to be like, where a lot of the investment attention in Silicon Valley and abroad is focused on systems approach to biology, right? I think people, I don't know anyone that would argue that it's, this is a very complicated network where any one intervention is going to cascade across, a number of endpoints. So where's the data? How far along is the ooplasm approach? Have you been working on cell lines, animal models, human models? How far out do we get to see this out there?
Yeah, so we are if we sort of divide up the company into really three parts today, number one, we are a U.S.-based biopharma development shop that has a 3-year program for the clinic in the United States. We have spent the last several years in the petri dish, in animal models, mice, rats, guinea pigs, some cats. And we have been moving forward sort of biotech development 101, but this is the United States and things take time just in terms of everything that needs to be done to get into the clinic here. But we've been moving forward specifically focusing on oncology models, the kidney, which is a major target of ours, and then the central nervous system and looking at the dynamics of both dedifferentiation and redifferentiation in vivo models. Secondarily, while we are not a consumer packaged goods company per se, needless to say, that is a huge industry in its own right, and we have a lot of interest and hence partners that are coming to the table that are interested in the non-RX opportunities today in terms of skincare because of the rejuvenation, regeneration angle. And we've been active in research on that front, much more in the petri dish originally, but then, the unique, idiosyncratic nature of that business, you have the ability to move to human testing quite quickly and have done a lot on the skin, looking beyond sort of efficacy, but at a lot of the things you have to deal with when you get into the cosmetic business. We are also very active in sort of traveling around the world and learning about other regulatory systems because, if you can get a product into clinical development 2 years faster in other countries nowadays, you have this really unique dynamic occurring, as I'm sure you're aware, with regard to the globalization of medical training and research. And when you see things like, Harvard Medical School now operating in Dubai, or Weill Cornell Medical Center in Qatar, and Newcastle University in Malaysia, it's a really global integrated system of R&D now. And so we are out there learning, talking to hospitals and other research partners and saying, what are the possibilities in the 200 plus other countries out there? Because, we're US based, we love the US, but Can
we jumpstart the process a little bit quicker?
Where do we jumpstart the process? Yeah, exactly. So these are sort of the three pillars of what we're about. We're moving as fast as we can in terms of Bio 101. It's just the nature of the business.
Yeah. Absolutely. I'm just curious. I mean, are there just cool anecdotes with your animal models that you could share with our listeners here? I mean, maybe I'm asking for like a freak story, but are we regenerating rat eyes or rat arms or how far along on the animal models are we?
No, we don't do anything in ophthalmology, but we have done a lot of I know these models are kind of nasty, but the traumatic brain injury models in rats and guinea pigs, and the animal cruelty people don't like this stuff too much, but you end up hitting the poor little animals in the head with a standard big weight drop model, and there's control groups and there's drug groups, and you study the brain and the sort of the growth morphology, and then at the same time,
are you having them run through like maze? Are you doing maze solve times?
Water maze testing, swimming, and so forth. A lot of that comes into play, and not only that, but in things like traumatic brain injury, there's a nice crossover with Alzheimer's because beta amyloid and other related neurofactors upregulated in brain injury. So you get to study sort of a wide range of gene expression. We've had some pretty promising results in those models. The cancer work is equally fascinating in terms of just how tissue remodeling occurs and how sort of over time you see sort of the what's described nowadays more of the sort of the cellular competition dynamic about how if you have a mixture of cells, how one group can outcompete the other and kill off the other, which is it's a very new area. Diego Moreno's group in Spain is a very major thought leader in the area, but it's really fascinating. I mean, this is what's occurring in our bodies right now as we're speaking, and how tissue that begins to go wrong or exhibit the wrong properties gets if they shut it up, and other cells in the microenvironment kill it. This has been, two rather fascinating areas. A side anecdote, in which we haven't really delved into too much, but are the some of the original work in brain removal, which goes back to the 1970s at University of Indiana. We haven't recapitulated any of that yet because it's been sort of off of our radar. But people forget that frog experiments were done at University of Indiana with salamanders, and actually complete brain removal is one of those things that sort of got us looking in the area of sort of regeneration in humans.
Right. It sounds like what One Flew Over the Cuckoo's Nest, or are those books you're just like, just lobotomying people.
Yeah.
Okay.
And here you have a case of pulling the brain out entirely, and little salamanders don't die. But it's really fascinating stuff. A lot. Once again, it's one of the sort of things I spend my spare time on. Sort of reading up on the history of what came before. But it's just really fascinating what nature can do when stressed. Let's put it that way.
I'm curious in terms of getting your thoughts on. So if these newborn animals and certain animals have these peptides in the ooplasm that are so helpful, why does nature have these disappear as humans turn into adults?
It really disappears much earlier than that.
Okay.
It disappears, after the first couple weeks of your conception. Why don't we? I get the question, why don't we have any complex regenerative capabilities? I think most of people in the evolutionary biology world will say, well, during that sort of reptile/mammalian transition that occurred, however, tens of millions of years ago, and when we became such major bleeders, and we're a species that bleeds very rapidly. We die very rapidly from loss of blood. When that happened, we, from an evolutionary perspective, became very good at laying down scar tissue, and very good at fibrosis and thrombosis. And sort of evolution went that way with us. But we do still reprogram. I mean, that is people talk about sort of cancer and the immune system all the time. It's not really the immune system that keeps cancer development at bay throughout our lifetime. It is as I mentioned before, the cellular competition in those microenvironments. When cell A somewhere in our body starts to go wrong and starts, expressing certain oncogenes, it's cell B, C, D, E, and F that sit around it that tell it to either calm down or kill it. And it does this not by, doesn't give that cell 10 milligrams of drug X. It secretes a whole set of paracrine factors that say, get into shape or we're going to eliminate you. And so we do have some of this reprogramming ability, just not highly complex. So obviously can deal with single cells, but limbs, no. We don't have that.
Yeah, I mean, I think with evolutionary biology, I think it's always very theoretical, right? You can't really, I think that's like the critique on that area of inquiry. But it seems to stand to reason that you have a local optima around, okay, scar tissue is going to let, bleeding out animals survive longer. And, the evolution doesn't have a goal. It's just, if there was any goal, it's just passing down genes and as part of natural selection. So that doesn't mean that we're optimizing for Jeffrey Wu or Ira Pastor or you listener out there to live forever. It's optimizing how you pass your genetic material to the next and sustain that. Cool. I'm actually curious about scoping out. I mean, it sounds like very promising pipeline you have there, both on the RX world and in the consumer world. But it seems like, from what we've seen in our community and just the broader discussion, there's clearly a growing attention with world leaders around the possibility, the promise around enhancing humans. And this dives into your work with the World Economic Forum. It seemed to be a relatively new council, and it's what is that experience like? Is it how real is that? Is it more on the theoretical? Let's talk about this, or is this like, hey, should we actually start talking about policies that government should be considering for regulating this field.
Yeah, it's a lot of talking and think tank type stuff, a lot of conferences and conference call discussions about you know, all of the issues, social and economic, ethical, that are going to be coming up, whether it is biotech related or whether this has to do with the energy or agriculture or everything else that affects us as humans. So yeah, I mean, it's fascinating discussions. Whether there's anything completely actionable in the short term that's coming out of it, not yet. But the fact that it is a target of such an organization, and that they realize that the genie's been let out of the bottle, and this is happening on many different fronts in many different forms. I think it's a good thing. And there's a lot of interesting people at the table from very different backgrounds. Hospital systems in India. You have people from AARP, from the Gates Foundation. They're bringing together a really broad constituency of thinkers to talk about these issues. It's a good thing.
Yeah, yeah. And then broadly, what technologies out there are you most excited about? In terms of one, I think people talk about CRISPR a lot, but how what is your sense on the broad scope of technologies out there? I think ketosis is interesting from just from a lifestyle perspective. We're working with ketone esters, which jumpstarting ketosis is interesting. I mean, in your experience and in what you've seen out in the world, what excites you?
I'm very I'm very interested in all lifestyle issues and all lifestyle changes. Before doing this, I spent several years in the phyto medicinal area, and I'm still to this day a very big proponent of natural products and sort of the incorporation of anything into our daily health and wellness routines that has to do with the natural world. We had the first 100 years an industry still that is based heavily on plants, bacteria, and fungi. But when we see some of the things that are happening, whether it be with the microbiome, whether it be with the virome, or these new topics, interkingdom signaling, how a living signal from one species can actually affect the genome of another, semiochemical communication. All of these areas, I think, are sort of another dimension to the natural product sphere that are extremely important for us to keep in mind. And the interesting with the microbiome work, which I find fascinating, is that it is an example of a case where, hey, the drug, as I mentioned before, in 2018 or 2025, whatever, is going to look different. And here, they're talking about drugs, which are in essence cocktails of living, bioactive materials in terms of that. The other area that I'm quite interested in, and I'd like to see how far it goes, is the area of so-called electroceuticals. My former employer, GlaxoSmithKline, recently invested, I think it was $75 million in this area, where they're basically saying, look, let's see if we can get the drug out of the equation entirely. The drug is just a chemical, and it's just sort of a stupid substance that doesn't really do much except transfer information between point X and point Y. And can we interfere without the drug substance? And the reason this is so interesting to me is that, if you and I were to go back 100 years in a time machine and say, go to some biology lab, you would be hearing all about that type of stuff because we didn't really know about molecular biology and other things. We'd be hearing about physics, forces and fields and all sorts of this interesting nomenclature that is we don't hear about nowadays in biology labs.
Yeah, it reminds me of college physics.
Exactly. Yeah. And now you have groups like, Mike Levin's set up at Tufts University and bioelectricity, which, is doing fascinating work in this whole area of, altering membrane potentials. He recently took, a frog and with electrical signals, he took the tail and he turned it into an eyeball. He talked about creepy things, but so
not just using electricity.
Yeah, just using electrical signals.
That reminds me of some of my friends work with TDCS or transdermal transcranial direct current stimulation.
Sure.
So there's, yeah, I mean,
All this stuff is pretty unique and fascinating to me.
yeah, so I think like that seems interesting in terms of like just seeing where I think it's in this space is like how do you disambiguate the hype from what is real, and hopefully as we see more and more data being published on that. So I'm actually curious to dive into it a little bit. Literally, skin cells are transformed into like I guess eye cells using electricity.
I think he created an eyeball. Like he morphologically, it was a this whole sort of biomembrane, bioelectrical, signaling is a more of a physiological dynamic. It's not controlled at the level of the genes. So I mean, he created an actual eyeball. Now I don't remember. I have to look back at the paper to see if the eyeball could see. That's a different thing. But I mean, so the fascinating thing to me beyond that is that, whether you're talking about electricity or magnetism or light, the effects of broader physical forces on gene expression, whether it be the astronauts that's hung out up there in the space station for a year and have all these biologic changes due to microgravity and solar radiation. I mean, it all is part of the big picture. And so I just, I enjoy seeing how sort of the micro and the macro level dynamics come together to achieve some of these types of outputs.
Are any of these interventions making it to your own personal routines? I mean, if, I'm curious, are you looking at intermittent fasting as something you do daily, or are you living a life, pretty standard Western, lifestyle? I exercise. I do all the basic stuff. I'm a big proponent of natural products. So I have my own, from my previous company, I have my own sort of routine. I'm big on cinnamon and turmeric and a lot of sort of the common spices that have years of peer-reviewed literature and very unique mechanisms of action that current drugs just don't work with. So I think those are legitimate in sort of daily supplement purposes. Fasting, that'd be like telling me, if you tell me the answer to life extension, living 1,000 years is to stop having sex, then I don't think I'm going to go down that path. So fasting is just one of those things that I love food. Not that I need a nasty Western style diet, but I just like to eat. So fasting is fair. I mean, I think the outcome is not just years of life; it's also quality of life.
Exactly.
I remember seeing there's a discussion around. There's a recent paper published studying Korean eunuchs and showing that the eunuchs had 10, 12, 15 years extended life than their equivalent counterparts that weren't castrated, leading to the hypothesis that testosterone, might not be optimal for longevity, and should people consider castration, if to the extreme, to extend longevity, right? And it's like I think, and then it's like the balance, like okay, does one optimize for years of life in that scenario, or is it a quality of life? That's a big part of it. And of course, there's quite severe effects of being castrated beyond just sex. I mean, it's such an important signaling factor for both men and women, but especially for men.
The whole dynamic of psychogenomics and the ability even to think and positive outcome affecting gene expression, that one is off the table. Would be off the table for me, needless to say. There's a benefit sort of just to being happy. And I'll leave it at that. The only other thing
I agree with that as well.
I wanted to mention based on your last question is I really do think that we need to do a lot better in terms of living based on our evolutionary understanding, and whether that is, we are humans, we have a human genome, but we contain the genes of all species that came before us, and all the stuff that kills us at some point, yes, was good for other organisms, whether that's high blood sugar levels or inflammation or even cancer. In terms of the a billion years ago was the reason, I guess, single cell organisms first got together to make multicellularity. But clearly, we need to live based on our evolutionary understanding. And if you were drinking 6 Cokes a day and going out of our way for large amounts of inflammation and whatever we do, it's not good.
Yeah, we gotta talk to the president about having his Diet Coke routine reduce a
Yeah.
little bit.
Yeah.
Awesome. How can our listeners follow your work or get involved? I mean, it sounds like there's a bunch of different threads, either by following Bioqark or there's ways to contribute to conversation with, think tanks. How do people get in touch and get involved?
Yeah, I mean, contact us on the website. We are a very open company. We are very transparent about everything we're doing, and get in touch. We disclose a lot of what we're, our research collaborations, the different things we're active in, and use the website. Get in touch directly with me if there's any questions. I love talking about what we do. I'm passionate about it, and I believe strongly in it. And the one message I like to get out there is that. Although this industry, and though I'm not in big pharma anymore technically, but I'm still part of the system per se, although, we've been seen as boring or evil or always promising things that may be 30 years away, we are at a very unique time right now where if we do things the right way, we are going to be translating a lot more human opportunities and affecting the human condition sooner than most people think. So it's a very positive future. I just want to get that point across.
Absolutely. We'll leave it at that. Thanks so much, and I hope to have you back on the program and have updates as we're curing diseases and allowing people to live happier and longer. Thanks so much, Ira.
Thanks so much for having me.
Yeah, pleasure.